9.5 Polysaccharides. The acid-insoluble pellet also has polysaccharides, carbohydrates as another class of macromolecules. Polysaccharides are long chains of sugars. They are threads, literally a cotton thread containing different monosaccharides as building blocks. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide, that is glucose. Cellulose is a homopolymer. Starch is a variant of this but present as a storehouse of energy in plant tissues. Animals have another variant called glycogen. Inulin is a polymer of fructose. In a polysaccharide chain, say glycogen, the right end is called the reducing end and the left end is called the non-reducing end. It has branches as shown in the form of a cartoon figure 9.2. Starch forms helical secondary structures. In fact, starch can hold I2 molecules, iodine molecules, in the helical portion. The starch iodine is blue in color. Cellulose does not contain complex helices and hence cannot hold I2. Diagrammatic representation of a portion of glycogen. Plant cell walls are made of cellulose. Paper made from plant pulp and cotton fiber is cellulosic. There are more complex polysaccharides in nature. They have as building blocks amino sugars and chemically modified sugars, example glucosamine, N-acetyl galactosamine, etc. Exoskeletons of arthropods, for example, have a complex polysaccharides called chitin. These complex polysaccharides are mostly homopolymers. 9.6 Nucleic acids. The other type of macromolecule that one would find in the acid insoluble fraction of any living tissue is the nucleic acid. These are polynucleotides. Together with polysaccharides and polypeptides, these comprise the true macromolecular fraction of any living tissue or cell. For nucleic acids, the building block is a nucleotide. A nucleotide has three chemically distinct components. One is a heterocyclic compound, the second is a monosaccharide, and the third a phosphoric acid or phosphate. As you notice in figure 9.1, the heterocyclic compounds in nucleic acids are the nitrogenous bases named adenine, guanine, uracil, cytosine, and thymine. Adenine and guanine are substituted purines while the rest are substituted pyrimidines. The skeletal heterocyclic ring is called as purine and pyrimidine respectively. The sugar found in polynucleotides is either ribose, a monosaccharide pentose, or 2' deoxyribose. A nucleic acid containing deoxyribose is called deoxyribonucleic acid DNA, while that which contains a ribose is called ribonucleic acid RNA. 9.7 Structure of proteins. Proteins, as mentioned earlier, are heteropolymers containing strings of amino acids. Structure of molecules means different things in a different context. In inorganic chemistry, the structure invariably refers to the molecular formula, example NaCl, MgCl2, etc. Organic chemists always write a two-dimensional view of the molecule while representing the structure of the molecules, example benzene, naphthalene, etc. Physicists conjure up the three-dimensional views of molecular structures while biologists describe the protein structure at four levels. The sequence of amino acids, that is the positional information in a protein, which is the first amino acid, which is the second, and so on, is called the primary structure, figure 9.3a, of protein. A protein is imagined as a line, the left end represented by the first amino acid and the right end represented by the last amino acid. The first amino acid is also called as N-terminal amino acid. The last amino acid is called the C-terminal amino acid. A protein thread does not exist throughout as an extended rigid rod. The thread is folded in the form of a helix similar to a revolving staircase. Of course, only some portions of the protein thread are arranged in form of a helix. In proteins, only right-handed helices are observed. Other regions of the protein thread are folded into other forms in what is called the secondary structure, figure 9.3b. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to the tertiary structure, figure 9.3c. This gives us a three-dimensional view of a protein. Tertiary structure is absolutely necessary for the many biological activities of proteins. Some proteins are an assembly of more than one polypeptide or subunits. The manner in which these individual folded polypeptides or subunits are arranged with respect to each other, example linear string of spheres arranged one upon each other in the form of a cube or plate, etc., is the architecture of a protein, otherwise called the quaternary structure of a protein, figure 9.3d. Adult human hemoglobin consists of four subunits. Two of these are identical to each other. Hence, two subunits of alpha type and two subunits of beta type together constitute the human hemoglobin HB. 9.8 Enzymes. Almost all enzymes are proteins. There are some nucleic acid that behave like enzymes. These are called ribozymes. One can depict an enzyme by a line diagram. An enzyme, like any protein, has a primary structure, that is amino acid sequence of the protein. An enzyme, like any protein, has the secondary and the tertiary structure also. When you look at a tertiary structure, figure 9.3d, you will notice that the backbone of the protein chain folds upon itself. The chain crisscrosses itself, and hence many crevices or pockets are made. One such pocket is the active site. An active site of an enzyme is a crevice or pocket into which the substrate fits. These enzymes, through their active site, catalyze reactions at a higher rate. Enzyme catalysts differ from inorganic catalysts in many ways, but one major difference needs mention. Inorganic catalysts work efficiently at high temperatures and high pressure, while enzymes, which is a biological catalyst, get damaged at high temperatures, say above 40 degrees Celsius. However, enzymes isolated from organisms who normally live under extremely high temperatures, example hot winds and sulfur springs, are stable and retain their catalytic power even at high temperature up to 80 to 90 degrees Celsius. Thermal stability is thus an important quality of such enzymes isolated from thermophilic organisms. 9.8.1 Chemical reactions. How do we understand these enzymes? Let us first understand a chemical reaction. Chemical compound undergo two types of changes. A physical change simply refers to a change in shape without breaking of bonds. This is a physical process. Another physical process is a change in state of matter when ice melts into water or when water becomes a vapor. Basically, here bonds are broken. These are also physical processes. However, when bonds are broken and new bonds are formed during transformation, this will be called a chemical reaction. For example, BaOH whole twice plus H2SO4 gives BaSO4 plus 2H2O is an inorganic chemical reaction. Similarly, hydrolysis of starch into glucose is an organic chemical reaction. Rate of physical or chemical process refers to the amount of product formed per unit time. It can be expressed as rate is equal to del P upon del T. Rate can also be called velocity if the direction is specified. Rates of physical and chemical processes are influenced by temperature among other factors. A general rule of thumb is that rate doubles or decreases by half for every 10-degree change in either direction. Catalyzed reactions proceed at rates vastly higher than that of uncatalyzed ones. When enzyme catalyzed reactions are observed, the rate would be vastly higher than the same but uncatalyzed reaction. For example, when CO2, which is carbon dioxide, combines with water, that is H2O, in the presence of carbonic anhydrase enzyme, it forms H2CO3, which is carbonic acid, which is a reversible reaction. In the absence of any enzyme, this reaction is very slow with about 200 molecules of H2CO3, which is carbonic acid, being formed in an hour. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction speeds dramatically with about 6 lakhs molecule being formed every second. The enzyme has accelerated the reaction rate by about 10 million times. The power of enzyme is incredible indeed. There are thousands of types of enzymes, each catalyzing a unique chemical or metabolic reaction. A multiple chemical reaction, when each of the steps is catalyzed by the same enzyme complex or different enzymes, is called a metabolic pathway. For example, when glucose converts to 2-pyruvic acid, C6H12O6 plus O2, it gives 2C3H4O3 plus 2H2O, is actually a metabolic pathway in which glucose becomes pyruvic acid through 10 different enzyme catalyzed metabolic reactions. When you study respiration in chapter 12, you will study these reactions. At this stage, you should know that this very metabolic pathway with one or two additional reactions gives rise to a variety of metabolic end products. In our skeletal muscle, under anaerobic condition, lactic acid is formed. Under normal aerobic condition, pyruvic acid is formed. In yeast, during fermentation, the same pathway leads to the production of ethanol, that is alcohol. Hence, in different condition, different products are possible. 9.8.2, how do enzymes bring about such high rates of chemical conversions? To understand this, we should study enzymes a little more. We have already understood the idea of an active site. The chemical or metabolic conversion refers to a reaction. The chemical which is converted into a product is called a substrate. Hence, enzymes, that is proteins with three-dimensional structures, including an active site, convert a substrate, that is S, into a product, that is P. Symbolically, this can be depicted as S2P. It is now understood that the substrate S has to bind the enzyme at its active site within a given cleft or pocket. The substrate has to diffuse towards the active site. There is thus an obligatory formation of an ES, enzyme substrate complex. E stands for enzyme. This complex formation is a transient phenomena. During the state where substrate is bound to the enzyme active site, a new structure of the substrate called transition state structure is formed. Very soon after the expected bond breaking or making is completed, the product is released from the active site. In other words, the structure of substrate gets transformed into the structure of product or products. The pathway of this transformation must go through the so called transition state structure. There could be many more altered structural states between the stable substrate and the product. Implicit in this statement is the fact that all other intermediate structural states are unstable. Stability is something related to energy status of the molecule or the structure. Hence, when we look at this pictorially through a graph, it looks like something as in figure 9.4. The y-axis represent the potential energy content, whereas the x-axis represent the progression of the structural transformation or states through the transition state. You would notice two things: the energy level difference between the S and P. If P is at lower level than S, the reaction is an exothermic reaction. One need not supply energy by heating in order to form the product. However, whether it is an exothermic or spontaneous reaction or an endothermic or energy requiring reaction, the S has to go through a much higher energy state or transition state. The difference in average energy content of S from that of this transition state is called activation energy. Enzymes eventually bring down this energy barrier, making the transition of S to P more easy. 9.8.3, Nature of enzyme action. Each enzyme E has a substrate S binding site in its molecule so that a highly reactive enzyme substrate complex ES is produced. This complex is short lived and dissociate into its product or products P and the unchanged enzyme with an intermediate formation of the enzyme product complex EP. The formation of the ES complex is essential for catalysis. The catalytic cycle of an enzyme action can be described in the following steps. Number 1. First, the substrate binds to the active site of the enzyme fitting into the active site. Second, the binding of the substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. Number 3. The active site of the enzyme, now in close proximity of the substrate, breaks the chemical bonds of the substrate and the new enzyme product complex is formed. Number 4. The enzyme releases the product of the reaction and the free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. 9.8.4 Factors affecting enzyme activity. The activity of an enzyme can be affected by a change in the conditions which can alter the tertiary structure of the protein. These include temperature, pH, change in substrate concentration, or binding of specific chemicals that regulate its activity. Temperature and pH. Enzymes generally function in a narrow range of temperature and pH, figure 9.5. Each enzyme shows its highest activity at a particular temperature and pH called the optimum temperature and optimum pH. Activity declines both below and above the optimum value. Low temperature preserves the enzyme in a temporarily inactive state, whereas high temperature destroys enzymatic activity because the proteins are denatured by heat. Concentration of substrate. With the increase in substrate concentration, the velocity of the enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity Vmax, which is not exceeded by any further rise in concentration of the substrate. This is because the enzyme molecules are fewer than the substrate molecules, and after saturation of these molecules, there are no free enzymes molecules to find with the additional substrate molecules, figure 9.5. The activity of an enzyme is also sensitive to the presence of a specific chemicals that bind to the enzyme. When the binding of the chemical shuts off enzyme activity, the process is called inhibition and the chemical is called an inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme, it is known as competitive inhibitor. Due to its close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzyme. Consequently, the substrate cannot bind and as a result, the enzyme action declines. Example, inhibition of succinic dehydrogenase by malonate which closely resembles the substrate succinate in structure. Such competitive inhibitors are often used in the control of bacterial pathogens. 9.8.5 Classification and nomenclature of enzymes. Thousands of enzymes have been discovered, isolated, and studied. Most of these enzymes have been classified into different groups based on the types of reaction they catalyze. Enzymes are divided into 6 classes, each with 4 to 13 subclasses, and named accordingly by a 4-digit number. Oxidoreductase or dehydrogenase enzymes which catalyze oxidoreduction between two substrates S and S dash. Example, S reduced plus S dash oxidized to give S oxidized plus S dash reduced. Transferases enzymes catalyzing a transfer of a group G other than hydrogen between a pair of substrate S and S dash. Example, Sg plus S dash gives S plus S dash G. Hydrolases enzymes catalyzing hydrolysis of ester, ether, peptide, glycosidic, carbon-carbon, carbon halide or PN bonds. Lyses enzymes that catalyze removal of groups from substrates by mechanisms other than hydrolysis, leaving double bonds. Isomerases includes all enzymes catalyzing interconversion of optical, geometric or positional isomers. Ligases enzymes catalyzing the linkage together of two compounds. Example, enzymes which catalyze joining of carbon oxygen, carbon sulfur, carbon nitrogen, phosphorus oxygen, etc. bonds. 9.8.6 Cofactors. Enzymes are composed of one or several polypeptide chains. However, there are a number of cases in which non protein constituents called cofactors are bound to the enzyme to make the enzyme catalytically active. In these instances, the protein portion of the enzyme is called the apoenzyme. Three kinds of cofactors may be identified: prosthetic groups, coenzymes, and metal ions. Prosthetic groups are organic compounds and are distinguished from other cofactors in that they are tightly bound to the apoenzyme. For example, in peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen, heme is the prosthetic group and it is a part of the active site of the enzyme. Coenzymes are also organic compounds, but their association with the apoenzyme is only transient, usually occurring during the course of catalysis. Furthermore, coenzymes serve as cofactors in a number of different enzyme catalyzed reactions. The essential chemical components of many coenzymes are vitamins. Example: coenzyme nicotinamide adenine dinucleotide NAD and NADP contain the vitamin niacin. A number of enzymes require metal ions for their activity, which form coordination bonds with the side chains at the active site and at the same time form one or more coordination bonds with the substrate. Example: zinc is a cofactor for the proteolytic enzyme carboxypeptidase. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in the catalytic activity of the enzyme.